Good afternoon. My name is Juexin Wang from Electrical Engineering and Computer Science Department and the Bond Life Science Center at the University of Missouri. I will open my own lab at the Department of Biohealth Informatics, School of Informatics and Computing, Indiana University, Purdue University, Indianapolis. My talk is about neural relation inference to learn long-range allosteric interactions in proteins from a molecular dynamics simulation. Many protein functions are regulated by specific dynamic biomolecular processes, such as allosteric, protein folding, unfolding, and protein activation. Allosteric ligand binding affects the active site remotely by spatially long-range intra-protein communication. However, it is very challenging to identify these communications. We propose NAR-MD model, Neural Relation Inference Molecular Dynamics model. It takes the trajectories of residues in 3D spaces from the molecular dynamics trajectories. This framework modeling the dynamics in different time stamps. It uses encoder to learn a distribution and reconstruct the trajectories. The learned distribution can be used to infer the edges between the residues. Based on that, we can get the interaction graph. In the case study, we use APO-PN1 with molecular dynamics simulation to study inter-domain allosteric regulation. APO-PN1 is a typical therapeutic target system which regulates cell proliferation, immune response, and treated as a drug target. Some known mutations as I28A weaken inter-domain interactions and reduce catalytic activity. In the analysis, we systematically explore 3 scenarios of the system. We run molecular dynamics simulations on each of the scenario. We can see the WW domain and the PPI domain does not interact together as the first scenario. In the second scenario, with the ligand binding, these 2 domains interact together. In the third scenario, with the point mutation, isoleucine mutated to aniline at the 28th residue, these 2 domains do not interact. The red line are molecular dynamics trajectories, and the blue lines are predicted trajectory from the NAR-MD model. We can see NAR-MD model reconstructs the trajectory well in these 3 scenarios, meaning it captures the intrinsic relations between the residues. Here are the edges learned from an NAR-MD model. Up line are residue level of the edges, bottom are domain levels. We can see the huge differences from the interaction graph we learned. In the beginning, WW and catalytic loop, the most important domains, do not have edges. With the binding, they have edges, and the edges disappeared in mutations. These phenomena are consistent with biological prior knowledge. Here is the interaction graph in domain-level illustration based on the learned edges with the directions. Here is the interaction graph from the sender to the receiver, and the receiver back to the sender. With the inferred interaction graph, we can get the inter-domain allosteric communication pathways by just calculating the shortest distances between different residues using the classical Dijkstra algorithm. With the binding, we obtained the whole pathway from start point in WW domain to the end in catalytic loop domain. When we have the mutation, we can see the pathway interrupted at the 28th residue. In summary, the NAR-MD model is one of the first attempts to use graph neural network to analyze molecular dynamic simulations. It can effectively generate the interaction graphs related to proteins slow motion through an embedding of reconstructed molecular dynamics trajectories. It accurately estimates for free energy changes across. It can reveal the pathways mediating allosteric communications. It can also be useful for studying other long-range, long-time interactions in molecular dynamics simulations. For example, pinpoint function-related driving residues in the conformational transition of amyloid beta peptide, and we are working on that. The code of NLMD is also publicly available at GitHub. I want to thank my advisor Professor Dong Xu at the University of Missouri, Professor Weiwei Han at Jilin University in China. Dr. Jinxuan Zhu, the co-first author, did not boast of the molecular dynamics works, and the paper was published at the Nature Communications months ago. Thanks to the funding agency NIH. And as an incoming assistant professor, I'm recruiting PhD students who are interested in deep learning and graph modeling in molecular biology. Come to me if you have any questions and if you are interested. Thanks for your attention.